So again, I'm just going to talk to you about two types of seaweeds that I really enjoy working with. I know City is very familiar with marl and rhodolith beds. These are a red algae, a red seaweed. They embed calcium carbonate within their cell walls. So that gives them this very tough cell wall, kind of like what you would see on a limpet. It's like an outer skeleton almost. And they're usually free living. And the way we distinguish between marl or rhodoliths is literally just through the branching. So marl we often think as being more ornate, branched kind of like this picture here, and rhodoliths are more like a pink rock, free living, still photosynthetic organisms, and they're pictured in this background screen throughout the talk. I'm also going to talk to you about kelp forests, focusing on the kelp forests that you see in the UK, but also Ireland. That's where I'm doing my work. And these are often, like City in her introductory slide had the giant kelps. So you often think about these huge kind of like marine forests that you can see from the sky. And what we have in Europe is actually more of a shorter species that is, I almost kind of like to talk about it as chaparral, like a chaparral system that you'd see in the Mediterranean, terrestrial habitats. It's gorgeous. It's about 1 to 3 meters in height, and it forms these dense communities along our shallow coastlines. So I'll be talking about that as well. And throughout the talk, I'll be presenting my own research, and I'll try and minimize the amount of graphs that you guys have to see. But I'm trying to kind of describe whole systems. So hopefully, if you guys have any questions, just put them up in the comment box, and I'll try and address them as fast as possible. So in my past work, I've worked on rhodolith beds and marl beds in Greenland. And these, again, are very productive habitats. The marl themselves are photosynthetic, but they actually don't produce that much oxygen. They may be more important as like a habitat. So while they're low lying and it looks kind of like a cobble bed, they actually have a very deep, they can be very deep, and that provides a very nice habitat for juvenile organisms in fauna that you can kind of see here. This is just a bunch of brittle stars kind of raising their arms to collect food. In this picture I think this is from marlin. So in Greenland, I worked on a species called Lithothamnion glaciale. It's pictured here, but you can also see this up in Scotland. It has a very kind of temperate to polar distribution within Europe. And in Greenland, it's predicted to be kind of found along the whole coastline. So if you look at the green crosses, it goes up past Cisco Bay, all the way down to the southernmost point and around to the eastern coastline of Greenland. And I was really focusing on looking at this species with some researchers at the University of Glasgow and Aberdeen. We were looking at using this as a proxy for changes in temperature and salinity over time. So they call it rhodochronology because it's a red algae, and you can create a chronological record using its kind of tree rings that grow within its thallus. And we were really interested in using this along with paleontology and glaciology to recreate the advance and retreat of a large tidewater glacier just outside the capital of Greenland, Nuuk. But interestingly, we did a ton of surveys in this major fjord system. So the Tidewater Glacier, if you can see my arrow, is up here around this bend. This is called the Ice Fjord, and it feeds into 3 major fjord branches here. This is a very big, productive salmon fishery here. And then here's a capital. And interestingly enough, we did a ton of drop camera surveys and scuba surveys. This species isn't really found. Lithothamnion glaciale, our target organism, isn't really found in this major fjord system. We're only seeing it in 2 locations within this peninsular area with high flow, and within this smaller fjord system, an area that also has very high flow. So hard to access on scuba, but also there's tons of other coral and algae in this area. It's just kind of interesting from a perspective of what is a specific species, what's unique to this species that's driving its distribution, that it can't tolerate being in a habitat like this that has a lot of high melt in the summer. So you have a lot of freshwater, cold freshwater input in the summer season, moving and flushing out of this major fjord system. So we ran a lot of experiments, some mesocosm study, you can call it. We've recreated 4 treatments here where we have a natural marine treatment, which is actually it's about 7 degrees Celsius and 33, salinity of 33 during the summertime. So this is again when you have the high melt. And then we've manipulated to temperature, dropping it and dropping the salinity to reflect high melt. So a temperature of 4 degrees Celsius, which is actually pretty cold, and 22, salinity of 22. We've done this as a 2x2 factorial, so you manipulate temperature and salinity independently and together to see what's really driving shifts in physiology. And if we look at seaweeds, they're not like animals, so you can't look at a behavioral response to different environmental conditions. But over a year, and especially in the summer season when they're supposed to be the most productive, right, because they have actual sunlight, polar seaweeds are kind of interesting because they can live in a habitat that has very little light through a long period, for a large part of the year, right? So from late autumn to early spring, sometimes no light in the subtidal marine environment. So very high, long periods of light conditions in the summer. So if we look at their ability to calcify, so embed calcium carbonate within their cell wall and produce oxygen throughout summer months from May through May through August, I believe, or March through September, you can see that in the marine treatment, you have higher calcification during summer when you have light, higher oxygen production. When the salinity gets lower, you lose that uniform signal. The species seems disrupted. You have very low oxygen production or negative, so they're actually becoming maybe heterotrophic organisms, net heterotrophic, meaning that they're consuming more oxygen than they're producing. Low temperature, more productive actually, so it's a polar adapted species. Calcification rate's pretty constant. And then if you look at the fjord conditions with the low salinity, low temperature, oxygen production decreases through the summer, but their calcification signal is really off. So this species actually seems to not be able to tolerate low salinity, and that may be driving its absence in this major fjord system. So what I can say about this project is that we were not able to use it easily to recreate this kind of temperature salinity proxy for this marine system. But we were able to kind of also go back and look at, okay, biodiversity of these habitats. Nobody's really been able to focus on these habitats. Diving in this region is actually very complicated because you have to wait for slack tide, and that's about a 40-minute window that you can get in and out of the water, do your surveys. It's also very cold, so you can't spend a ton of time in there. But we were really interested in, okay, so if Lithophamian glacial is not found in this area, and we were really expecting it there, there's this Clathromorphum species, which is a rhodolith forming species that's pretty dominant in these areas, these kind of channels in the seaward islands off of Nuuk and also within the peninsula. And we know that there's a lot of kelp beds. This is a Saccharina longicruis species. It kind of dots the coastline here. But nobody really described the productivity or the diversity of both the rhodolith or the kelp beds. So what we did is we used our drop camera surveys, and we kind of went to 4 different sites for each type of habitat. Did some video transect surveys, meaning that we took a video camera. It's very cold, so we couldn't necessarily go around and count everything within a swath of habitat on one dive. We took a video camera and we did a video survey on a swath of habitat, analyzed that video later for just basically counting the number of different seaweeds, invertebrates, fish that we saw in each habitat per unit area. That allows us to really calculate biodiversity. We also took pictures of a unit area of the benthic habitat, so we were able to calculate what is the abundance of certain organisms within rhodolith or kelp beds. And then we took cores of each habitat. So I said, marl beds form this really intricate structure that's not necessarily above ground. It's like a nice matrix in the benthic environment. So we're really looking at whether or not rhodoliths have higher biodiversity than kelp beds, which are either found on bedrock. So you can't core bedrock, obviously, but the quadrats would look at benthic biodiversity in that way. But they're also in this area, they're really interesting. They can be free floating or persist on just small cobbles. So we were able to take sand cores and look at infaunal diversity within kelp systems. And we were able to compare that. So once we did that, we kind of brought everything back to the lab in Nook, analyze biodiversity that way, and I'm going to show you 3 different types of results to demonstrate the difference between the habitats. So this is a multi-dimensional scaling plot. What it does is it just compares how related each sample is. And again, our sample data is abundance of different types of species. So it takes that information and it just plots the site or the sample in relation to how alike it is to the other habitat types. So if we have kelp forests are the asterisks and rhodolith beds are these circles, you can see they cluster apart kind of nicely. The rhodolith beds are slightly more alike than the kelp forests are. The kelp forests are slightly disparate. So this is survey data. And what we found is that the common species that you see in kelp forests are going to be barnacles. That's interesting, right? Many of our barnacles in Ireland and maybe the UK are intertidal, so you never really see them subtidal, but there's probably really high flow in these systems. So they're able to feed and persist in these subtidal habitats. We have a lot of amphipods in kelp forests. The purple urchin that actually is found to graze down kelp forests in Norway was very common. And then we had a lot of sea cucumbers, so this is a filter feeder. And the rhodolith bed, sorry, this is a bad label. We had a lot of barnacles, but actually this system was mostly described by the huge abundance of the purple sea urchin. So again, 10 times more purple sea urchins and rhodolith beds than you see in kelp forests. It's good for kelp because they like to eat kelp, but it's also interesting in the sense that do these urchins kind of regulate fleshy algal sediment in this habitat, allowing this rhodolith bed to kind of persist. And of course, if you look at these habitats and we looked at the number of coral and algae versus the number of kelp in marl versus kelp beds, they're each defined. Their defining organism is obviously most abundant, but there are kelp within rhodolith beds and there are marl, but also coral and algae within kelp forests. So they're not mutually exclusive at all. If we look at what's covering the bottom, so we take these quadrat data and we look at that. Again, kelp are the asterisks and rhodoliths are these circles. They partition out really nicely. And what the defining species in these systems, for kelp forests, you're going to see bedrock is about 60% of the bottom. There's a lot of coral and algae on this bedrock themselves, as well as kelp holdfast. In marl, or in rhodolith beds, you have rhodolith, but also a lot of urchins covering the system. So again, just another way of defining and describing habitat differences. If we look at cores for the rhodolith beds, the rhodolith beds and the kelp forest, again, the kelp forest cores have to be done in sand. They also partition out quite nicely. So you have your asterisks and you have your circles kind of over here. And what you have in these systems with kelp forests, you're seeing a lot more mobile crustaceans in these cores, whereas with marl beds, you're seeing bivalves, juvenile echinoderms, so things like sea stars, urchins, brittle stars, a lot of chitons, so a lot of smaller organisms, juvenile organisms, and that might be important because this worldwide marl beds are kind of known to be a rookery for fish and other invertebrate species. Okay, so moving on, I mean, along with larger, the biodiversity of larger organisms, we're also really interested in the microscopic community. And this work has not been published yet, so I'm just going to briefly go through it, not too in depth. But City and I actually went to a conference in Roscoff where I presented this material. So we're really interested in bacterial communities, but other microscopic eukaryotes, even funguses, and how they contribute to the rhodolith community. So like I said before, with the low salinity in the marine environment, oxygen production really went down on Lithothymnian glacial, right? It became almost like an animal. So it was consuming more oxygen than it was producing. And so I was really actually interested in how these species kind of function and how much that microbial community that lives on Lithothymnian glacial contributes to its productivity, to its ability to kind of persist in really harsh conditions that can be found in polar habitats. So that's not just the low light, but there's also very low nutrient availability in these habitats. So are nitrogen fixing bacteria an important contributor to this system? All sorts of stuff like that. So the way we kind of targeted this question is we use microsensors, which is pictured here, to look at oxygen production across a variety of light levels in a transect from the surface of the individual, so where the nice biofilm community is found, to the bulk water column. And we do that with oxygen, with nitrogen, and we did it on two different types of seaweed. So we looked at the natural rhodolith or marl with the nice biofilm, and then we kind of created an axenic culture, meaning that we removed all the bacteria and the eukaryotes from it, just to look at the variation in its physiology. We also sequenced the microbiome of these individuals, but also of the water column across the fjord system, and also from the surface of lots of other seaweeds in the habitat, because we were really interested in defining the natural environment or the natural microbiome community on seaweeds, but also in the environment, and then how that might change in these mesocosm conditions that we had kept Lithothymnian glacial in a previous paper that I described. So this is just a map kind of showing you where we took all of our samples. So you can see we sampled freshwater runoff from the glacier, down the ice fjord, down Godthap's Fjord, which is just outside of Nook, and then from seaweeds at the upper fjord system, in the peninsula, and in this minor fjord system. It's a big research base. We also sampled this fjord system right here, which does not have a tidewater glacier, but also has a lot of runoff through this this valley here, just to kind of look at different environmental conditions and what bacteria live there. So again, we also sample our marl after they've been in these mesocosm treatments, looking at how environment might shift the microbiome on seaweeds. So just for some preliminary data that I can show you, we have this NDS plot again, and you have all of your different colors showing you the site within the fjord, and all of your different shapes showing you the type of seaweed that they're on. And I think the most distinctive thing you can see is that the communities that you see in the water column are very distinct from the communities that you see living on seaweeds. And that's important because seaweeds are, one, benthic, but also two, may actively regulate their biofilm community. So they produce oxygen, but also a lot of other compounds that could be dangerous or what we call allelopathic for bacteria or other microorganisms. But also, if you look at communities on seaweeds, I'm not going to show this data necessarily, but they're much more diverse and much richer, meaning there's more species on the seaweeds. If you look at the shapes, you can see that Phaeophyta has a very big mix. Rhodophyta is kind of clustered over here by itself, and we had very few green algae, the Chlorophyta, but they're kind of mixed in there with the Phaeophytes in terms of their community structure. So rhodophytes were very unique. Their communities were very unique in this sense. If we look at our mesocosm communities, there's a few outliers here that we have multiple different substrates, which I won't necessarily talk about, but you can see that the marine communities, low salinity communities, and then fjord and low temperature kind of communities all cluster out together pretty distinctly in this NBS plot. So the environment is really shaping these bacterial communities on these seaweeds, which is, I think, is very interesting, and hopefully we'll get that data out there soon. So I'm just going to show you a bit of oxygen production using microsensors. Again, so we're looking at that oxygen production on natural individuals with their whole microbiome community, but then axenic individuals, so individuals that are just marrow, nothing else. And if you have no light in the system, they're naturally producing no oxygen, they're uptaking. So it's a net uptake of oxygen. That makes sense because without light, you can't really do photosynthesis and produce oxygen. But they're producing, or they're taking up more oxygen in the axenic communities. So without the biofilm, they use up more oxygen. If you increase that light to a very low light level, you start having positive production. And again, this is in two regions. Sorry, I should have said this. If you look at your marrow over here, you have this interbranch area and you have an outer branch area. So that's just showing you the two different regions. Within the biofilm, you have positive production of oxygen at very low light levels. And then this goes up when you increase to a higher light level. So the biofilm is producing a lot more oxygen than the actual branch tip in these natural individuals. Across all light levels, you basically have net oxygen uptake in axenic individuals. And here, the biofilm location, this is just an interbranch location without any other microorganisms. So kind of an interesting display of how that biofilm is actually contributing to photosynthetic production in marrow beds themselves. So the whole community is really important for providing that structure in the marine environment. Okay, so now I'm going to really shift to kelp systems. And I kind of got interested in this. Juliet Brody wrote this nice review of how marine communities may shift across these different regions. So from the Lusitanian region, so southern Europe, all the way up to the Arctic, how they may shift with future ocean conditions, specifically future high CO2. And so if you look at the Arctic, you see a reduction in the number of marl beds, an increase in kelp beds in future ocean conditions. There's also a shift, in the boreal and the Lusitanian region to less marl, less coral and algae, more invasive species. So this is an invasive kelp here, Undaria pinnatifida, or non-native, however you prefer to use it. And so I was really starting to get interested in, okay, so what are future Arctic communities going to look like, as well as boreal or Lusitanian communities in Europe. And so I started a fellowship in Ireland with the Irish Research Council, basically just to create a modern baseline for marine ecology. And I actually moved to Ireland right when, I'm not political by any means, but I moved to Ireland right when Trump got elected. And there's a shift, I think, worldwide towards a more conservative perspective in politics, but also maybe a reemergence of this kind of news cycle that didn't take fact as for what it is. So I like this quote from Dr. Jane Lenchenko. She used to be the head of NOAA under the Obama administration. Now is the time for a quantum leap into relevance. And as we tackle these emerging challenges, and she's referring to in natural history, but also in marine science and ecology in general, don't forget to carve out time to connect with nature and people so we can recharge our batteries and remind us of what's important. And maybe this is even super relevant in terms of what's been happening during the pandemic, our kind of isolation from communities, and our ability to kind of maybe reconnect with the natural environment. And when I moved to Ireland, I would have liked to ask a lot of questions about, chemical ecology within kelp forests, or, how they might look in future ocean conditions. We didn't really have a basic understanding of what was present, how productive these systems were, how they were functioning, and where they were even found. So I had to focus on creating a modern baseline for kelp forests, which is kind of what we had to do in Greenland when we realized our target organism wasn't there. And so in Ireland, our kelp is actually Laminary hyperborea. It's also called cuvee or mayweed, maybe tangle in the UK. It's this lovely species. It can be up to 15 years in Ireland, I think 18 years in Norway. So it's a cold water species, loves it. And you can age it by kind of cutting the base of its stipe and looking at the rings within it. So kind of like tree rings, almost similar to marrow species. So it's very long-lived, and it has a northern distribution in Europe. And this is just a picture from Jorge Cease's paper in 2016. This species has been present for a long time. So in the last ice age, its distribution was restricted to southern Europe and the Mediterranean. And as that ice age eased, it's moved northward up to its most northernmost point is in Svalbard, I'd say. But now what we're seeing is that it's restricting its southern distribution. So it's disappeared along the southern coastline of Portugal, and it's moving northward as the oceans kind of warm. And so it's kind of interesting because there is potential for us to start losing these habitats if the oceans warm too much. And that's just what this bottom image here is showing. This is a projection for the worst case scenario from IPC 2013. And obviously this scenario is actually potentially looks worse in newer IPCC reports. So the main thing is that we don't know what we have in the system, so we can't really monitor what we're losing in terms of looking at IPCC projections or anything like that. So the idea here was to create a baseline for kelp forest ecology, create a monitoring program that can contribute to worldwide networks on kelp forest ecology, but also things like conservation programming, MPA programming, which Ireland is really starting to move towards, and just future research projects. But then the last thing that we were really hoping to do is understand the resilience of these habitats. So their ability to bounce back from extreme events and recover their natural function, whether this be, maintaining their trophic cascade within the ecosystem, maintaining productivity. So to do this, it's actually quite complicated to do this because these systems are below the lowest astronomical tide line. So to do this, we had to set up subtidal monitoring buoys that we could relocate both data loggers but also transects for monitoring these systems. We were really interested in what is moving into these habitats and settling. So we set up recruitment habitats using two different types of structures. And what this does is it captures either pelagic larvae, so a lot of invertebrates have a pelagic life stage within their life cycle, and that could also be true for fishes. But we also have this at the bottom to look at what is recruiting from the local community. So what's staying, what's reproducing and restocking the local community. We also looked at succession plots. So this is how the community recovers after a large disturbance event, like a storm moves through, boulders kind of turn over in the subtitle. We have biodiversity transects. These are basically swaths where we count everything like we had done with the videos in Greenland. We looked at biomass of the kelp itself and the diversity and population structure of that kelp. And we looked at growth of that kelp to understand how fast it was growing and how productive it might be. So if we look at growth, this is a species that likes to grow in the winter. So it's kind of contrary to other kelp species. So we have an intertidal dominant laminary digitata or oarweed. We have Saccharizia polychaetes or fir bellows, all these other species that tend to grow more in the summer. And that's when they reproduce and stuff. But this dominant is actually more as a winter grower, and it also reproduces in the winter. So it starts reproduction, and then it starts growing a new blade in January. And this blade has peak growth from February to March, where it can grow up to a centimeter a day. That's pretty fast, not as much as the giant kelp, which can grow a meter a day, but I think it's pretty fast for how big these individuals are. And if we look at biomass, which is important from maybe an applied perspective, if you were to harvest these individuals, but also from a resource perspective for the whole community, it's a food source. We see that biomass kind of peaks around 2 to 4 meters depth. And then we actually see the oldest individuals also in that area, which is interesting. And it starts to slow down as you get deeper in the water column. And that kind of makes sense from a, kind of light perspective. So they're getting less light, maybe not able to grow as much, but also maybe a disturbance perspective, or they're just getting outcompeted at those depths. What's interesting is that for these kelp forests, the denser they are, so the more individuals you have per meter square, generally the younger they are. So they actually kind of self-thin, like you might see in a natural forest, where older individuals kind of shade and outcompete younger individuals that are moving into the habitat. So when you find the oldest individuals, if you have your median age of 6 or 8, you have far fewer individuals per meter square. So it's kind of cool. It's working like you might see a terrestrial forest work. And then if you look at the kind of sugars that you can find within the kelp tissue, and this is important from a food web perspective, you have laminarin and mannitol, and the concentration of these within tissue kind of shifts throughout the year. So you have higher mannitol concentrations summer and autumn, these decrease in winter and spring, and laminarin then increases winter through spring. Across ages, this actually varies quite a bit. So you have peak laminarin in kelps from 3 to 5 years old. Mannitol is fairly steady until you get very, very old, around 9 years old, and then mannitol and laminarin both peak. So this is interesting if you think about density of older individuals, how that population density decreases. I think there's definitely implications for the amount of sugars provided for the communities. And then if you look across site, there's high variation. So site-specific characteristics, the environment, is going to dictate how many sugars are provided. So this is very interesting from like a food web perspective, and we have a master's student from Finland who's creating a food web model so we can better understand these dynamics within Irish kelp forests, but also kelp forests across the EU. Okay, so now I'm going to show you data from biodiversity. or biodiversity data. So I wanted to show you how important kelp forests are as a habitat and how distinct they are from other shallow habitats. So that is, here's your kelp forest number A, but you also have these bedrock communities here that have like high sponge, high acidian, sea squirt densities. They, these are like walls that you might see in sea locks or on exposed shorelines. We also have sediment bottom cobble communities that are very common, like for instance in Kilkerran Bay. Loch Einn, like the eastern coastline is very sediment dominated by sediments and so on. We have marl beds like this one here, and then we also have this lovely biogenic reef. It's a sapurlid worm reef. And so fairly unique among European coastlines. It's only really found in Scotland and Ireland, and maybe one location in Italy. So just wanted to show you how kelp forests kind of are distinct in that sense. Here you have your different shapes defining a habitat type, and you can see that kelp forests really cluster out. They're most alike, more alike than any other habitat. Your sapurlid worm reefs are here, and you have the sediment bottom and bays. Everything kind of clusters out. So each habitat has its own unique community throughout the year. And even within kelp forests, we do see some interesting partitioning across site. So there are site-specific characteristics that define biodiversity at each location. And just so I can define biodiversity in kelp's ecosystems, what really stands out is the number of echinoderms. So that's everything from this common sea urchin, which is not necessarily going to overgraze kelp forest communities. This is an omnivore species, so it eats detrital kelp, but it doesn't actively eat the live kelp. You have a lot of cucumbers, juvenile sea stars. Generally, this is a common star. This is a spiny star. A lot of mollusks, so the purple and the gray top shell. And then seasonally, we see a lot of juvenile gadoids in the water column. You can sort of see them. They're very tiny. So in this picture, you can only see a few here and there. But we also see a lot of crustaceans. So quite a lot of common or edible crab, spiny crab, and then the lobster as well. And this is pretty, I think that a lot of people already know this, right? Because you see fishermen putting their pots inside kelp forests for crustaceans, from April through July. So seasonally, we see a lot of peaks in this. Okay. If we look at what's kind of choosing to settle in these kelp forest habitats, we have two types of settlement substrate. This larger one was intended for fish that have a pelagic life stage, and the smaller one was intended more for invertebrates that might settle in the area. And again, they're at two different heights in the water column, one to capture the pelagic cohort, and one to capture the local kelp forest recruits. We didn't really see any difference in the recruits across depth. And that may be because kelp forests are found in pretty shallow habitats, so there wasn't a huge partitioning between those two locations. But that also could be because there's not a lot of fish with a pelagic life history stage in Europe, or for Ireland specifically. Sorry, and then the invertebrates are not, necessarily selectively choosing between either of those habitats. So what we see is there's a lot of seasonal patterns in recruitment within these structures. So we're looking here at data from our smallest habitat type, and this usually captured a lot of Nemertean worms, a lot of crustaceans, and a lot of bivalves. But we see some partitioning across seasons, showing that there's seasonal pulses in different types of larval recruitment. And that's also true for the bigger habitats. The issue with this kind of stuff is that in winter and spring, you have these massive storms coming through Ireland. Okay, let's also say in summer and autumn, you do too. So it ends up taking out a lot of your habitats. And this is the problem with marine ecology in general, is that the environment often dictates your data collection, whether you can go out, but also whether your things stay in the water themselves. So we ended up losing a lot of data, but we do see seasonal partitioning here. And in this, these larger habitats, the seasonal differences are going to be driven by your crustacean recruitment. We have a lot of crab and lobster larvae kind of pulsing from spring through summer. And that's interesting because you see more crustaceans then. Now, these seaweeds don't only provide a habitat, but they are a habitat themselves. And that's what kind of is interesting and unique about this species. Other species of kelp like oarweed or the golden kelp, which is slowly moving northward, don't have a lot of things living on either their stipe or their holdfast. So if you look at the size of the stipe, this increases with age. Your diversity and your richness in your species assemblage increases with mass, so increases with age. This is also true for holdfasts. So the older the individuals, the more diverse they are, and the more species there are found on them. And I think that's really important if we're thinking about wild harvest of these, is to figure out a way to leave individuals, especially older individuals, in those habitats, because they are themselves a habitat, not just a food resource. And when we looked at succession, again, this is just recovery of the system after a disturbance. We actually have native plots where we left the coral and algae in place, and then we put out these pavement tiles that were sterile, theoretically. They were just concrete, so they needed conditioning in the marine environment. If we look at recovery of these systems, so we put the sterile plot in at the same time as clearing this substrate, what we see is a lot of juvenile kelps kind of coming back within 100 days of clearance. Like, that may not seem very fast, but it's actually pretty fast for a microscopic organism to grow to a macroscopic. After a while, we start seeing thinning of the population. Just pretend like some of those juvenile kelps got deleted until they start growing. So they're self-thinning. Again, same thing that we saw as the older the average age is for a unit area, or for the kelp forest community, the fewer individuals are there, right? If we look at the sterile succession plots, we start seeing a turfy, maybe biofilm community forming where sediment can get stuck into after about 40 days. We start seeing invertebrate settlement after about 115 days, more invertebrates, larger invertebrates after 140. And then after 340 days, we start seeing kelp settlement. So the kelp has had a full year, or these have had a full year in the situ to get conditioned. And obviously the kelps have to have a reproductive event in the winter to actually settle on these systems and then start growing. So recovery in ecosystems happens much faster on natural substrates. And this is actually what we're looking into now, is that there's this very interesting life cycle within seaweeds, well, within kelp specifically. Seaweeds have a variety of different life cycles that it's just too much to talk about right here. Within kelps, you have a large sporophyte that we're all very familiar with. And the sporophyte has 2 pairs of chromosomes, much like our body, right? Their cells undergo meiosis, meaning that they divide to create they divide to create zoospores. And this is kind of the same thing that happens within our tissues to create both eggs and sperm. And these zoospores settle in the bottom, and they become gametophytes, which are independent microscopic stages that produce both the female stage, so the eggs, and then the male stage, so like the sperm, male gametes. These recombine on the female and then become the sporophyte. So there's a whole independent stage here that we don't really study very often, that may be responsible for quick recovery in native plots. So we've cleared these plots completely of all macroscopic species, sponges, kelps, all that. But these microscopic stages are allowing recovery really quick. And so that's what we're investigating right now in our kelp res project that's funded by the EPA. So we're looking at resilience of kelp forests, and that includes where have laminary hyperborea forests been found historically. Trying to go back to the 1700s, we were able to recreate records. So this is just a rosary from Donegal, made out of, actually made out of oarweed stipes. But there's a lot of data kind of in these historical records showing you maybe how species distributions may have shifted over time. We're really interested in the genetic diversity, and we've published a paper on this, showing that the highest diversity is actually found in the Southwest. And that's interesting because if you think about the historical distribution through the model that was created by Jorge Zis that I showed kind of at the beginning of the kelp section, kelps had always persisted in Southern Ireland, Southern UK, Brittany, Portugal, through the last ice age. So it might be a glacial refuge for the species and theoretically should be more diverse than other regions that have been recolonized since that ice age. So we're really interested in the connectivity across populations. If you look at the life cycle, the mobile stage for the sessile species, the species that doesn't move, is really within the zoospores, and then really only with the male gamete seeking out the female egg. So the dispersal potential is very low, and they may not have the chance, adjacent populations may not have the chance to recombine at all. So we're looking at connectivity, genetic structure across Ireland, and maybe mating system variation in these species. Then we're also looking at natural resilience in the life cycle. So looking for banks of microalgal kelp forms, whether the zoospore, the gametophytes, or maybe the juvenile sporophyte can remain dormant and allow this very quick recovery of the system after disturbance. And then we're creating future monitoring tools that includes scuba surveys, which I'm going to ask you to do on the next slide, but also maybe using this pipeline to assess kelp forest distribution with satellite data. So I hope you guys can keep your eyes peeled. A lot of our information is distributed through Twitter. Our site is at KelpRes. So I just want to end this talk first with our QR code. This would lead you to, if you're a snorkeler or if you're a scuba diver, if you wanted to go visit a kelp forest either in Ireland or the UK, if you go to this link here, use this QR code, you'll find a quick kind of survey asking you, did you see this kelp? Did you see that kelp? So what species were found within your kelp forest? Asking you to estimate density, asking you then to estimate maybe density of the common sea urchin, density of other species that are really kind of came out, like popped out when we did our diversity surveys. So we're trying to create a metric to assess indicator species that indicate healthy kelp forest ecosystems in this region. And then I just want to give a shout out to Sea Search Ireland. There isn't really a great scientific diving community in Ireland, so none of this work would have been possible without Rory Antonio Callahan, who runs Sea Search Ireland in Galway. So they basically create a lot of our species records for the subtidal across Ireland with other dive clubs, and they're the only reason we know what we have in these habitats currently. So I like this quote from Joni Mitchell just to demonstrate how important that is. And if you would like to support them, this is their GoFundMe page. They're pictured here in Lettercallow, which is an amazing sapropelid worm reef that they've just gotten funding to continue monitoring. So basically these two have day jobs, but they do this all of this work in their spare time. and they're integral to any kind of research that's happening in a subtidal in Ireland. So this is just a quick acknowledgement slide. It was funded by a lot of folks, and these are all, well, not all of my collaborators, but quite a few of my collaborators, mentors, and students that have helped facilitate this research. And then lastly, I'd just love to answer any of your questions if you guys have any. And I know there were 2 questions actually in the chat, so I can kind of address those first if you'd like. Mhm. That's a good question. So, we have a point in time measurement of urchin density. Um, it'd be very cool if we could monitor their movement throughout the day because they are very mobile species. We see them much more prevalent in rotolith beds, and this has been hypothesized, or this has been observed, in other systems, so in Norwegian systems as well. And, one thing that could be happening is that, maybe kelps are settling in rotolith beds and we don't know. But the urchins are so dense there that they're just grazing down and precluding this forest from forming. And you also have to keep in mind that, as I mentioned, these rotolith beds were found in channels, in the seaward islands of Nook as well as in the peninsula. So there's a lot of high water flow. And that also might be precluding kelp forests from forming in this region, but also could be beneficial for urchins. They have a lot of tube feet that collect particulate matter in the water column. And so they could be feeding on other things besides kelp. Well, they most likely are. They're not only eating kelp in kelp forests. And when they graze down kelp forests, they're still eating stuff after the kelp forests are gone. So they're pretty omnivorous, and kind of voracious eaters. So I think that they're probably feeding on a lot of different stuff. And, that's really something that we kind of would have to test, is whether they're precluding kelp forests for forming, if they're found there because of the current, what's really happening in these systems. But it is an interesting, it is a really stark difference in both communities. And I'm assuming that's in kelp. So if you look at this, the depth, sorry, if you look at the depth here, actually the median age is really kind of, for all sites and all, for all sites is going to be, if you look here, it's around 3 to 4 years old is the median age of kelps in this area. That doesn't mean that we don't find a lot of older adults. It just means that and I think that new individuals are recruiting into the bottom habitat annually, and so you get a lot of juveniles as well as a lot of older individuals. And there's nothing that is, there wouldn't really cut off density. This is kind of just what's the right word for that? It's just kind of like a sliding scale, I guess is the best way to say it. There's no cutoff. It's a sliding scale, which is kind of best exemplified by this beautiful slope, this negative trend line as you increase age with the, against density. There's another question from Shruti Joshi. Um, well, um, I think the main thing is just everybody involved because a lot of people like to use natural habitats, daily. So whether you're going for a walk or if you're going for a swim, it is very helpful for people to submit species records. In Ireland, we have the National University Data Centre for this. I don't know what there is in the UK for these citizen science records. Certainly, Sea Search is not just Tony and Rory. They train a lot of dive clubs to do their species records as well. But I think, if everybody contributes a couple species records, we better understand our environment across a broader range. I could go on. Funding agencies, especially like things like NERC and the IRC, but also NSF in the US, really need to start reinvesting in natural history science. We don't really understand. I think that we've done some really good work in a few systems, and then we just assume that all kelp forests are alike or all marine forests are alike, and they're just not. And we really need to better understand every system before we start manipulating it. And we can use that as nature-based solutions for climate change. And I think that's becoming more, people are starting to understand that more. And so we're moving more towards that, but it's a slow process as everything is. And hopefully, funding systems will pay attention to that. But mostly it's going to rely on citizen science. Let's just go with that first. So NBRC or something? And the NBRC? I don't know. I don't know that they are. I think that what we saw with Lithuanian glacial was like a very species specific response to that really fresh system. You'll notice if I went to the picture here, sorry, Disco Bay in northern Greenland up here is also very fresh, and you actually only find well, they say rhodoliths. I guess they haven't identified the species in this paper, but a lot of Clathromorphum around Disco Bay, and then you see Lithuanian kind of adjacent to that. So that seems like a very species-specific response for that one type of marl. In terms of rhodolith beds versus kelp forests, I really think that we need to invest more research time into that kind of system, because if we look at those in Ireland, they're kind of inhabiting different environmental conditions. So it'd be really hard to say, right? So like marl beds are generally going to be a little bit deeper. For instance, I think you found them right on the inward side of the Aran Islands. A little bit deeper where kelp wouldn't maybe naturally grow. And they generally don't like bedrock, which kelp really loves. But as your geology shifts, as current patterns shift, I think both distributions will probably shift. And then you might get a replacement. Certainly, I think you're going to have, if we look at this Brody paper, I think you're going to have with, warmer oceans that are more acidic, you're going to have a breakdown of that calcium carbonate wall, and that's why you're going to lose those rhodolith or marl beds up in the Arctic. It's very hard for organisms to calcify if the water is too acidic. It also shifts the saturation state of the necessary ions that they need in that water. And then kelps are, if it's polar, if there's more light, kelps are going to grow better.